anaerobic versus aerobic versus fermentation, metabolism of cells. We're going to talk about how cells generate ATP, which is energy. There's respiration, both anaerobic and aerobic, without air and with air, respectively, and then there is fermentation into alcohol, lactic acid, etc. Respiration is a three-step process: glycolysis, Krebs cycle, and the electron transport chain. Glycolysis is just the breakdown of glucose, the 6-carbon sugar. That can happen with or without oxygen, and glucose is broken down into pyruvate and also generates a bit of ATP. In the Krebs cycle, which follows next, there is oxidation of acetyl-CoA into carbon dioxide, also produces some ATP and some important reductive coenzymes like NADH and FADH, which we will use in the electron transport chain partially. In the electron transport chain, we're going to follow an electron through the membrane of the mitochondria. NADH donates an electron, and a proton is thusly pumped out through the dehydrogenase complex, the DH here. The electron goes through the Q-quinone complex into nitrate reductase, where then nitrate, NO3, is reduced by gaining that electron into NO2 minus, which is nitrite, and also a little bit of water. Typically, that's where the electron transport chain ends in the use of nitrate. It can continue on into a process called denitrification, where nitrite is reduced further into nitrous oxide, and then nitrous oxide is reduced even further into nitrogen gas, which is typically poisonous for cellular processes, and is not extremely common in cells, but it does occur. This would be the end of the electron transport chain that then goes to fuel ATP synthase to produce all of its ATP. Aerobic is exactly the same, except the final electron acceptor, which is the NADH in, final electron acceptor is nitrate, which is NO3 minus. In aerobic, it's just oxygen, and the end products are always carbon dioxide and water instead of nitrite and water. Next, we go on to talk about fermentation. Fermentation is part of everyday life if you are above 21 and you drink that much. But it partakes in creating alcohol. We have hops, malted barley, and wine as just a few examples, but it is a crucial bit of producing alcohol. It couldn't be done without it, because fermentation, at least ethanol fermentation, the end product results in the cell putting out alcohol, which is part of the consumption, part of our consumption. Hops have a special bacteria on them that does this, or contributes to this, and also puts flavor. Fermentation, the actual process of it, starts with glycolysis. Glycolysis goes the same. At the end of everything, it's glycolysis and then just fermentation. It's just those two steps. At the end of fermentation, the final electron acceptor that fuels than ATP synthase is some organic non-oxygen molecule. This is typically really inefficient. It only makes about 2 ATP versus 38 ATP in respiration. So it's inefficient, but it also produces carbon dioxide, which is helpful in things like bread. Also, some of these bacteria are helpful in creating yogurt, but they also contribute to food spoilage. So they take particular handling to work in our favor in the industry. But what about something dangerous to us, like Mycobacterium tuberculosis, that is the infective agent of tuberculosis? Well, it is actually found to be possible to switch its metabolic methods between anaerobic and aerobic. It is technically a prototroph, and it was found to either use sugar or use host-derived sugars, like sugar and its surroundings, or host-derived sugars, fatty acids, amino acids, cholesterol, carbon, etc., during stages of infection. Even when not infectious and it's non-replicating, it needs to make ATP to survive. The pathogen, in order to learn of ways to attack it via medication, was studied in differing oxygen environments. It was found that in a low oxygen environment, it has significantly reduced ATP production and a very high NADH to NAD plus ratio, indicating that the electron transport chain is not occurring. There's a buildup of NADH because there is no protomotive force happening. However, if grown on glucose, it can actually use the surrounding CO2 and do a reverse Krebs cycle and make a little bit of energy. It's not a lot, but at least it's doing something. It's important to know these kinds of things so that way experts can create medications that attack not only one form of metabolism for the cell, but all forms of metabolism for Mycobacterium tuberculosis and others.